First up is Leeza Santiago Millan from Harvard University discussing revealing the biology of long non-coding RNA through discovery of their interacting proteins.
All right, good afternoon, everyone. I am really happy to be with you guys today and to kick off the short talks. And so I like to start my talks with acknowledgments. And I feel like I always like to introduce the village behind my thoughts. And so first, I want to thank my lab, which is the Novena Lab. They took me up a year and a half ago, and since then, they've supported me a lot during a transition of labs. And I also want to give a special shout out to Urara, which is a co-lead in the project that I'm going to be presenting. I want to thank members of the Vidal Lab, because thanks to them, I'm becoming a high throughput scientist, my dissertation advisory committee, and people that I call the Brain Trust Brigade, because they basically drank my Kool-Aid and now they help me with my scientific thoughts. And so I want to start us off with talking about Shwachman-Diamond syndrome, or SDS. So within Dana-Farber, we have some doctors that they're specialized in being able to identify these patients and treat them. And so the symptoms in the patients start appearing from 4 to 6 months of age, but you can have an adult onset. And mainly what you're going to be looking for is skeletal abnormalities, pancreatic insufficiency, and a low number of blood cells, meaning they're going to have bone marrow failure. Interestingly, 5% of the children will end up developing leukemia. And if they make it into adulthood, the number is going to increase to 25%. However, why are SDS predisposed to leukemia? No one knows. The main cause of SDS is a loss of function mutation in the SBDS gene. And basically, this gene has a major role in ribosome biogenesis. It's not that you don't have ribosomes, but at the end of the day, it's kind of like a slower process, is kind of the gist of what people are observing. And so at the end of the day, when you have loss of the SBDS protein, you're going to have a global reduction of protein production. And that drives the second question in the field, which is how does global protein reduction lead to very tissue-specific abnormalities? If we take a step back and think about just general genetics, we're always taught that DNA is transcribed into RNA and then translated into protein. And that's very much true. To this day, most diseases are going to be filled with minds thinking about them, but those minds focus on proteins and the dysregulation of proteins. However, this RNA will only become protein around 2% of the time. The rest of the 98, we're going to stay in the non-coding RNA space, and within that, my lab really likes to think about the long non-coding RNAs. But then to keep in mind the context, I'm literally saying out loud, I like to study proteins. Which proteins? The really big ones. And that's where the field is at. Within the SDS patients, no one has ever considered whether or not there is a problem with the long non-coding RNAs. And so we took interest in that because they were showing such tissue-specific expression. And with the little that we know of the link RNAs, we have been learning that what's interesting about them is that if you find an essential function, it tends to be specific to a tissue. And so do link RNAs have a role in SDS pathogenesis? So the first thing that we did, just do a differential expression analysis. We took the bone marrows of SDS patients, we compare them with healthy individuals, and what we see here in the volcano plot is, so for anyone who hasn't seen it, if you look behind the 0, this is suggesting downregulated genes. Upwards of the 0, you're going to see upregulated. So there's definitely a dysregulation of the link RNAs, but the one that we want to focus on today is CRNDE. Why? CRNDE ends up being part of the 2% of RNAs, long non-coding RNAs, that have some research behind it. And it turns out that CRNDE is upregulated in a lot of different cancers, one of those cancers being leukemia. Additionally, because we saw this raise in leukemia, we went into public databases and we wanted to understand if CRNDE has any sort of relationship or value as a prognostic factor. And it does. So not only are you going to find that CRNDE levels are increasing in cancer, if you're looking at the levels of CRNDE amongst the patients, you can stratify and have a negative prognostic value the higher the level of CRNDE. All of that was correlation, so we wanted to really test out, is this something that we can model in the lab? Is it just a correlation? This is not really relevant to what we're doing. So the way that we wanted to observe this was with blood cell stem cells. And so basically here what we want to observe is, can the stem cells self-renew, and can CRNDE help them self-renew? So in this experiment, CD34 is used as a marker to identify the blood bone marrow stem cells. And so when we do an SBDS knockdown, so we're going to follow this black dotted line, we can see that we're losing the self-renewal capability of these stem cells. However, when you express CRNDE or overexpress CRNDE, we start to see a rescue. And again, keep in mind that the moment that we knock down SBDS, we're having a proteome level change. And the addition of one transcript is now making a rescue. And that is really hard to capture in lab. I'm going to say we're really happy that we're seeing this change. So all of this to say, what is the function of CRNDE? This welcomes you guys into our field of long non-coding RNAs. I was also presented to the problem with the lab. Lisa, what is CRNDE doing? Make a thesis out of it. All right, I come from a protein world. In the protein world, I like to look at conservations. But link RNAs in general have poor conservation, so that limits any alignment and that limits species comparisons. CRNDE has conservation with mice, and it actually has a relatively high number of publications, but to this day, there's no real mechanism behind what it's doing. The second thing we like to do with proteins is make structures out of them. But with long non-coding RNAs, they're more similar to mRNAs. So they're considered kind of like disordered RNAs. And it's not that they're disordered, it's that when you start modeling them, you don't get just one option. So it's a whole beautiful science trying to discern which of everything is biologically significant. And then lastly, we have to keep in mind that a lot of these link RNAs are tissue-specific expression and function, meaning if you go to the lab and you're not seeing the result that you want, you might be in the wrong tissue, and if you're in the right tissue, you might be looking at the wrong time. So how did I want to approach this problem? Literature has shown us that these link RNAs have DNA binding capabilities, RNA binding capabilities, but also protein binding capabilities. And so although these long non-coding RNAs are not really conserved, their protein binders are definitely conserved. And so if I can get an idea on who you'd like to talk with directly, I can start to have a map in my mind of where do you want to hang out, what type of music are you listening to, just show me who your friends are. I'm definitely going to get an idea about you. Now within the field, we definitely have RNA-protein interaction methods. You can do pull downs, you can do mass spec, but again, these can be really limited, really hard depending on the tissue that you're working with, and what we wanted to develop was something that the field in general could use. So we're definitely in need of a systematic, high throughput, unbiased approaches, because right now we're lacking. So the way that I wanted to approach it is with budding yeast. So I come from a yeast genetics lab, so I became a yeast geneticist at the beginning of my PhD. I love yeast because it is a tiny eukaryote. It is a whole eukaryote, which is my favorite part about yeast. It has rapid growth, it is economical, it has a suite of genetic tools, and it's very easy to adapt for high throughput screening, which is something that I definitely wanted to do. I am not reinventing the wheel though. I'm going back to the classics, and the classics is the yeast-3 hybrid method. So this method is reporter-based, and so what we're going to do is that we're going to have a promoter, and it's going to drive the expression of a gene called His3. Basically, the yeast need it to survive. If the reporter is on, you're good. If it's off, the yeast die. And then we're going to have 3 pieces that we can add into the system. This left side is not going to be changed, so we have a known protein RNA interaction with the MS2 coprotein and the MS2 stem loop. And we're going to fuse our RNA of interest to this MS2 stem loop. So this is going to make sure that our RNA is at that site. And then the MS2 coprotein is fused to a DNA binding domain that can talk to the promoter. However, these 2 pieces are not sufficient to turn on the reporter. You need your protein of interest fused with an activation domain to be at the site, and then you're going to drive expression. So because I wanted to make this high throughput, what I want to really see, I don't want to test in a specific tissue or bias in any way the type of interactions that I'm capturing. So if I have a non-coding RNA of interest, I want to search the whole proteome for its protein interactions. And so originally, we have this collection of yeast where individual yeast are housed with one individual plasmid with a unique protein. but this ends up being around 240 96 wall plates. You can't bring that into any lab, especially not if you're the one pipetting. So I wasn't about to do that. And I love to think about equity by design. And so one of the first things that I did is create what we call centipools, where now I'm pulling 100 proteins per well, but they're 100 individual strains. But I don't want to lose the sensitivity of my assay. So we do yeast tricks. So yeast beautifully can exist as haploids, but also as diploids. And so they're called MAT-A and MAT-alpha, but you can think about it as female and male. And so the MAT-A is going to be carrying our RNA of interest, and then the MAT-alpha is the one that's being pulled into 100 different strains. However, when you drive the mating between them, mating is a binary event. And so at the end of the day, the readout of the reporters is directly of one protein and one RNA. Then you just have to spot the diploids in the selective media. And the way that that looks like is kind of like this, where now we're going to see these individual colonies where a well had 100, but this individual colony signifies one RNA, one protein, and then through Sanger, you can get the identity out. You can also play with the stringency of the assay by adding a toxic compound, so 3-AT. And so all you need to understand about it is that if you have 3-AT, the yeast will definitely die. But then if you have history in the background, the yeast is happy. And so it's not that you're going to get false positive without 3-AT. You can think about it as low affinity interactions are captured in the absence of 3-AT. And then when you add the compound, you're going to bias towards really strong interactions. And so where we're at right now is that we ran the assay against CRNDE with and without the compound. Here we can see that when I added the compound, I'm having a lower amount of wells, which is expected, and the majority of them are coinciding with the promiscuous assay, which is exactly what you want to see when you run it. We were able to then get the Sanger back, and we have a list of 213 genes. So starting out with around 20,000, dropping down to 200, I'm a little bit excited. I had really hoped to have the identities for you guys like validated, but because I work and make mistakes, I'm a week behind.
Great job.
I'm a week behind. Yeah, but we definitely have a list. We're definitely seeing some interactions. The interactions look interesting and it looks messy, and I like messy, but it would make sense with what we think might be happening with CRNDE. So really today, the different things that I want you guys to take away, one is the existence of the SDS patients. They're a rare community, but we need to think about them. In general, there is this trend that bone marrow failure patients have a predisposition to leukemia. No one really understands what's connecting them. The second thing is that link RNAs are dysregulated in the bone marrow of these patients. So again, when you guys go back to your lab and you're thinking about any biological problem, please keep in mind that you have link RNAs in the background. That might be the key of whatever is missing or whatever you cannot make sense. I hope that I convinced that CRNDE, out of a lot of different dots, was an interesting candidate for us to focus on, because we could see that independently, if you have healthy people and then you look at the bone marrow failure, it's going down, the levels are going down, but then when you have healthy going into leukemia, the levels are going to come up. And so we can start to wonder, if we look at bone marrow SDS patients and we track them into their leukemia, will we see the levels of CRNDE rise? And why is this important? If the bone marrow failure patients get a transplant, which right now is the only main thing that they can do, they have an 80% survival rate. But as soon as they develop the leukemia and then get the transplant, it drops to less than 20%. So being able to identify early on when the switch is happening is really important. And that lastly, that using yeast to identify the proteins that can bind to this long non-coding RNA is a very, very brilliant approach. It's my thesis, so please support my thesis, to systematically discover the biological functions of any long non-coding RNA in the healthy states and then in the disease states of interest. And then I can take any questions.
Thank you for that. That was great. We have time for one question, unfortunately. Well, I was told to give it to students anyway.
I'm a student of one.
Go for it.
Oh, no, I think one of the students really should.
Yes. All right.
For that nice talk. I was wondering, at the beginning, you did some, you showed your volcano plot of your RNA sequencing. Have you looked at gene ontology enrichment analysis to look at what biological pathways are involved? Specifically, not just for CRD, but all your downregulated non-coding RNAs?
I didn't. I personally did not. No, we didn't look at gene ontology. We already had, so choosing CRNDE came before. We can't trace how there's a postdoc that left the lab, but he was the person that got the Eureka. But we can't even contact him at this point anymore, and so we can't trace back the idea. So we had to go to the differential, do the steps, and actually show that it's downregulated. But the moment that we had it, then we started running with it. But definitely, it's something that we will do and see if there's a broader pattern between. I'll also say that, just to throw that out there, when I looked into the long non-coding RNA papers, I noticed that when CRND is dysregulated, there's other very famous long non-coding RNA that's also dysregulated called hot air. Hot air is downregulated in bone marrow failure, but it's also downregulated in AML. So then something that really just continued to push us, and not necessarily looking at the pathways itself, but realizing that we might have caught some switch, and so we're just focused on the switch right now. But definitely something that's going to happen.
Thanks so much. You can